Good morning, everyone. I hope you're all safe and you're all doing well. Apart from having fun, I hope you're also learning a lot. So, in this time, because of this coronavirus thing, we have a set of challenges in terms of learning and teaching. It is a challenge for me to teach. Um, and I take these challenges as an opportunity to better my skills of presentation in on online. Similarly, I hope that you are all enthusiastic enough in to learn or take it as an opportunity to improve your learnability. In the sense, learnability is the skill to or ability to learn at a faster rate with minimal resources in a way. And the highest form of learning is, or the skill of learning is self-learning. It is a situation or it is a kind of empowerment where you can do it by yourselves without anybody else trying to explain you a concept in different ways. That requires a lot of motivation, but once you attain that skill, I think it will be a lot of help. So in today's lecture, I quickly put together, I went through some references, resources, and then I put together a slide to discuss about the molecular biology of coronaviruses. This is primarily because of the situation for two reasons. One is that we have to understand what coronavirus is or what is the, how does it transmit, how does it propagate and other things. One thing you should understand is it's just an RNA, and it's fantastic. It has, and that is our problem also, that it has a fantastic way of propagating very rapidly from person to person. And second objective of why I'm decided on this topic is it gives us a good platform to review whatever we have learned so far, and also that we will learn some of the exceptional molecular concepts that the coronavirus have. So if you can think of it in this way, excuse me. So if you can think, we will see how it goes. Sorry. So the first thing in this slide, in this lecture, we will learn a little bit about the introduction to the diseases, and then we will go to structures of coronavirus, and then we'll follow with the description of the genome, and then how it replicates, and then how it transmits, and then the precautions that we have to exercise so that we can control. And one more objective I have is to bring in an inspiration about the kind of research that you want to pursue in the future, what you want to do. So as you know, I think this is a little over somewhere around 2 days back, around more than 2 lakh people are affected, and I think as of today more than 10,000 people have died across the world, and that is a kind of problem that we have all over the world. If you notice in this case, over February and so on, so what you see is there is a dramatic increase in the during March. The number of cases has dramatically increased, and you can see that all over the world there are a lot of cases. And this is a problem that is now currently a global problem, which can threaten a large population of humans. So there are two terms that are typically used. One is called as epidemic, and the other one is called as pandemic. Epidemic is an outbreak of disease that affects many within a small geographic location, or it can be large. Whereas pandemic, if you compare, you can see that in pandemic, several locations are affected all across the globe, and then one can assume that the rate of killing will be much higher. So what we will also learn in this, we will learn further, is how coronavirus is able to do these things. Firstly, the coronavirus is a group of viruses. There are alpha, beta, gamma, and so on, different groups of viruses. The common feature is that they have a corona. Firstly, this is a picture of sun. The sun has a kind of, I hope you can see these flare-like things that are there around the sun, and those are referred to, that is referred to as corona. The whole outer circle that you see here is the corona around the sun. If we look at the coronavirus under electron microscope, we can see a similar kind of corona around it, and that's one of the reasons why it is called as, why this group of viruses are called as coronaviruses. So there are a lot of proteins on surface of these viruses which actually give these are the projections that look like corona of this virus under the electron microscope. So the internal structure or schematic diagram is well shown here. There are several proteins. Firstly, we have to understand that it is enveloped virus in the sense that it has a lipid bilayer all across. I hope you can see that. This is a lipid bilayer. These are the hydrophilic regions, and you have these hydrophobic regions. And there are several proteins that are there. The ones that is spike glycoprotein. I hope you can recollect what glycoprotein is. A protein to which an oligosaccharide is attached is called a glycoprotein. There are other proteins, hemagglutinin, acetyltransferase glycoprotein. There is a membrane glycoprotein, and there are small other enveloped glycoproteins and so on. These are the structural proteins that are involved in the structure of the virus. And then there is a protein called as nucleocapsid, which is nucleocapsid phosphoprotein that binds around the genome. What you will realize is that the genome of this coronavirus is RNA, and it is single-stranded RNA. And it is also message sense RNA genome. I hope you can recollect what is message sense. It means it is plus strand. Whichever has codons that are readable by the ribosome, that is similar to that of the virus, similar to that can be coded. Sorry for the interruption. Okay. So the message sense RNA genome, it is single-stranded and it is plus strand. That means it can be directly translated by the ribosome, and it has the spike glycoprotein, which is this one. Those are the ones that are essential for binding to the receptors on the host. Then you have hemagglutinin, acetyltransferase glycoprotein, and one of the important things that we have to learn about coronavirus is it is a RNA-dependent RNA polymerase. I hope you can recollect about the central dogma in molecular biology. DNA can give rise to another DNA molecule, that is by replication. It is mediated or catalyzed by DNA polymerase, which is a DNA-dependent DNA polymerase. And then we have transcription from DNA. You get RNA that is mediated by RNA polymerase, and RNA polymerase is a DNA-dependent RNA polymerase. And then we have from RNA we can get proteins that is mediated by ribosomes, and the translation is because from language of nucleotides we are now in the language of amino acids, the sequence. So this is a peculiar, interesting one because it is a RNA-dependent RNA polymerase. We will see in the next slides as we proceed. So if there are two aspects, two types of proteins. You can classify them if you want to. There are structural proteins. All these proteins are structural proteins. Those are important for the formation of the viral particle. And there are non-structural proteins which are responsible or essential for formation, replication, and other similar non-structural work. Here NSP is non-structural protein 3 and so on. And if one would look at the genome of coronavirus, it is the genome that is full length. It is around 30,000 nucleotides. It has two large ORFs, that is ORF1 and ORF2. Sorry, ORF1b. This is ORF1a and ORF1b. And there are small other ORFs of 2, S, M, N, and so on. S is for the spike protein, M is for the membrane protein, nucleocapsid protein, and so on. So the genome can be translated, and when it translates, it will produce two different large proteins. One is called as PP1A, polypeptide 1A, and there is another one, polypeptide 1AB. That is, if this is translated till here, you get PP1A. If this is translated from here till here, that is called as PP1AB. How can this be? The point is, we are already saying that this is one ORF and this one is another ORF. Then how can it produce a larger polypeptide like this one? We will see it in the next slide. It is because of a very interesting phenomenon that is referred to as ribosomal frameshift. So these proteins are again proteolytically autoprocessed to generate smaller fragments which are involved in two essential, or at least the essential functions, such as formation of the replication transcription complex, which is the one that are important for generating more copies of the genome, and also mRNA. We'll see it in the next round. So this part of the genome is mostly dedicated to production of structural proteins, and the ORF1A and ORF1B are mostly involved in mostly, not limited, mostly involved in formation of non-structural proteins that are involved for stability or generation of genomes and stability of genomes. So here is a model for coronavirus replication and transcription. The first thing is, this is the genome. You can say that the one that is given in red, that is the genome. And the ribosome translates and produces PP1A and PP1AB, and that forms the RTC, that is replication transcription complex, which will allow the formation of the minus strand. I hope you remember that this is the genome is plus strand. So from the minus strand, large amounts of the minus strand can be replicated by RTC complex, and will generate several full-length genomes, which can be used or packaged into new viral particles, such as something like this. There is also another thing that can happen. The same complex can also make shorter RNA molecules, such as these. These are, again, all the blue ones are in negative or minus strands. This is a minus, and then RNA polymerase will generate from this transcription of these subgenomic copies, will generate smaller mRNA like this, which are translated to produce structural proteins such as S, N, and so on. So quick review. This one is the genome that is in the red. It's full length. it is translated to produce the proteins PP1A and PP1AB, and these proteins generate minus strand, genomic and subgenomic minus strands. So genomic is full length, and the subgenomic are the smaller fragments such as these ones. The full length genomic one is translated, or you can actually call it as replication here, to produce full length copies of the genome. The subgenomic ones are transcribed to generate smaller fragments, and these are when translated they make a lot of proteins that are involved in which play a role in the structure of the coronavirus. So the interesting things that we understand here is about the RTC complex, replication transcription complex. The second one interesting part of it is the ribosomal frame shift. I was describing from ORF1A and ORF1AB or ORF1B. How can you produce two different proteins? So this is about description about the production of ORF1AB. I hope you remember that there was one ORF, and then there is ORF1A and there is ORF1AB, and the ribosome can translate to produce a very large 800 kilodaltons protein. How is this possible? So there is a concept or a phenomenon that is called as ribosomal frame shift. There are two. Say for example, I hope you can see this. Here is a codon UUA and then AAC. This is the ribosome and this is the mRNA that you are seeing here. The mRNA sometimes can form a pseudoknot. A pseudoknot is primarily intramolecular hydrogen bonding that happens, because of which the ribosome fails to move forward easily. So sometimes what may happen is this ribosome is actually moving in this direction, and sometimes because of obstructions such as these, a reverse or backtracking may happen. So it was actually reading, the ribosome was reading the AAC as one codon, and it has added the amino acid. We will read more about translation in the coming lessons. But because of this pseudoknot, it has moved back by one base. So it was reading AAC as one codon, but now it is reading AAA as one codon. And when it is moving, and then again it is actually by default trying to move forward. So when it moves, now it moves again, and it's now reading CGG as a codon. So what you should understand here is it has read C here already, and it is reading C as a part of another codon again, and those are referred to as -1 ribosomal frames. I hope you can see it here as well in SARS coronavirus. It's an example where UAA and then AAC. Here is AAC is the codon, and because of one frame shift, the ribosome, which has already read AAC as a codon, is now reading, it has moved back, and then it's reading CGG as a different codon. So C is read twice as part of AAC codon and as part of CGG codon. So there is also a positive +1 frame shift where the ribosome was reading UUU, and then it has moved further. It should actually read CGU as a codon, but instead it has moved forward as GUC, reading GUC as a codon. It has never read C as a part of any. It has just moved +1 base, and then it starts reading. Here it has moved one base backward and starts reading. So this ribosomal shift, it allows the reading of the ORF1A and ORF1B. So when the ribosome is synthesizing, assume that here is the ribosome 1. Here is until here, if it were ORF1A, and then here it is ORF1B is starting here, and just that if the ribosome can shift this reading frame, it can continue to produce the ORF1B part. So until here it translated one ORF1A, and here it can translate to ORF1B. That is how it can produce a large protein. So the replication is something interesting as well. So the coronavirus has the S spike. It will bind to certain receptors that are present on the cells. We will see it in the next slide. Here, let's focus on the formation or replication as a whole. So the virus, because it is an enveloped virus, it is likely to fuse the membrane and deliver the RNA into the cytoplasm. Then the host ribosomes, they translate and produce PP1A and PP1AB. And those proteins are proteolized and made into proteins, that is replication transcription complex, which will produce the negative strand, this one, and that negative strand is used as a template to produce the plus strand, multiple copies of this plus strand. That is genomic replication. There are also small parts of the negative one can be used, or the small parts of subgenomic transcription can happen, producing several structural proteins. These proteins are integrated while production itself. They are integrated into the endoplasmic reticulum, and then from the endoplasmic reticulum, they move on to the Golgi complex nearby. That is the endoplasmic reticulum Golgi intermediate complex, ERGIC, and the RNA that is produced full length is coated with the nucleocapsid protein, and then it is taken in, and then it forms a vesicle inside. It is processed. Oligosaccharides are also added there, and then finally the virus particle is released. While this is done, a large number of the host cell will die very quickly in some time after producing many of these viral particles. These viral particles again move on to the nearby cells, and that is how they cause a lot of damage to the host cells. So here is something, a comparison between SARS and the SARS-CoV-2, or that is the COVID-19. We had SARS epidemic some years back, around 2002, and now we have this COVID-19 variety. So they both belong to coronaviruses. There are differences, and but there are also similarities, because they are, and that is why they belong to the same thing. So there is something called ACE2, that is angiotensin converting enzyme 2, and all those cells that have this angiotensin converting enzyme 2 are targets for these viruses, SARS as well as COVID. And so if one is trying to have a therapeutic agent, then we will try to interrupt or not allow the viral particle to interact with ACE2. So there are two strategies. One is to make a compound that will bind to S protein, spike protein of the virus, so that it cannot interact with ACE2. Or there is another way of doing it, is to block on the ACE2. You have inhibitors. You bind those. You allow the binding of ACE2 to the inhibitor, because of which the virus cannot bind to the ACE2. That is another strategy. There is one more protein. There is TMPRSS2, which is a transmembrane protease, serine protease. which is which is a protein that is required. It's a host protein that is required by these viruses to process the S protein and other things. So if we can interrupt the processing of these structural or spike proteins, for example, of these viruses, then also we can stop the propagation or multiplication of the viruses. There could be several other ways of doing things. Say, for example, replication transcription complex is a good one because it is viral encoded, and if we can find inhibitors for it, then we can probably be able to control this virus. So people have developed some antibodies that can neutralize TMP RSS2, and they have done it for SARS. They have seen significant response neutralization efficiency. There is for COVID, but it is not as much. So the transmission, how it happened, is probably these are zoonotic, in the sense they are viruses of the of animals, which may or may not cause damage to those animals. Those animals might be co-evolving with or co-evolved a strategy of mutual survival somehow. But they may be, or we are probably very sensitive to these ones. Sometimes people, when they eat or handle live wild animals, through which these viruses people may come in contact with them, and those viruses, whichever is specific to our body, will infect us and cause a lot of problems. This COVID-19 has as I said, angiotensin converting enzyme is in our lung cells, and the virus infects those, because of which we have irritability and we have coughing and sneezing a lot. And the other thing is that because some of our the lung cells are dead, we have shortness of breath, in the sense because they are dead, the gaseous exchange is limited, and so we tend to the body tries to compensate by breathing faster, and that is what is referred to as shortness of breath. So what we should try to do is try not to have any contact with people. Try not to. The transmission is simple. Either when the person sneezes or coughs, those small particles released into air, which if we breathe, we might get into that. So the first thing is those who are affected should be isolated from other rest of the people. They should use masks so that whatever during coughing and sneezing, they are not releasing it to other people or even the medical personnel who are trying to help them. So avoid gatherings, that is where one sneeze or cough might actually mass infect a large number of people, and from then on each of these persons going to their homes and sneezing and coughing, you can imagine the amount of damage that can do to the population. So there are several ways of diagnosis. We also learned about PCR. We didn't learn about real-time PCR. Here is a PCR-based method. I'm not going to describe it, but the technique that is used is reverse transcriptase quantitative PCR. So they have sequence-specific primers for ORF1b. That is, they have a forward and reverse primer. That is one PCR. There is another test they do for N protein, which is a structural protein, a forward and a reverse PCR. So they take the swabs from the infected or the suspected cases, and then they because the genome is RNA, one has to convert that into DNA. That is where the reverse transcriptase comes. Reverse transcription is done, and upon that, the cDNA that is formed, PCR is carried out. Real-time PCR is the only thing is that you have the capacity to quantify the amount of product formed after each round of PCR, and that is quantitative PCR. Maybe we will learn this in a later some other time. In the future, we intend to have vaccination. Prevention is better than cure. So the idea is the body can synthesize antibodies, but if the synthesizing the amount of effective antibodies in quantity and quality is delayed, and before that the virus has multiplied much, then the person usually dies. And because it is affecting something like lung, which is an essential one for the gaseous exchange and for the whole system to run, the death can occur much faster than the immune response. So if we can prime the body to synthesize and keep the antibodies ready, and if we happen to have come in contact with this virus, we already have the antibodies ready, and which can hit out or eliminate the foreign viral particle. So the idea is, there are several strategies. The basic concept is somehow to make a protein directly injected into the body or through other methods, and then only the protein. We are not giving the virus. So this protein is a foreign particle, because of which the body will, it's not going to cause damage by itself, because it's only a protein. But the body senses it as foreign, and it will try to make antibodies against it, and that is how we can put it. That is how our immune system is primed, or it gets memory of that one and stores it, so that any future attacks happen, then it readily produces antibodies and then eliminates the viral particle. In the course, in immunology course, in the next semester, that is where you will learn more about this. Finally, this is the last slide. Stay safe. There are a lot of people, doctors and nurses and many other personnel, working hard continuously away from parents, and you should understand that they are quarantined along with the patients, and they work tirelessly and continuously. This should serve us as a motivation for our own careers of what we should do. As bioengineers and biotechnologists, we have a golden opportunity to pursue several things of how to develop diagnostics, for example, how to develop therapeutic agents for these things, and so on. We can make a lot of these cases, we can minimize, and we can actually play a role in the future and be ready for any kind of these things. I hope you will all be inspired, as I once told you about virology. If you learn about viruses, you will learn a lot of molecular biology, because a lot of concepts such as, for example, the frame shift, ribosomal frame shift, or replication transcription proteins. These are fantastic strategies these viruses have come up with, and they are the most efficient ones, and that's why it's important to study virology as well. You have as an elective, you will have virology in the future courses. If you are interested to pursue you should try to take it up. Be positive, stay home, and be happy, and make these very fruitful. Okay, thank you very much. Have fun.